It's like two soldiers. That's how they always get called. The one group of soldiers, one group of T cells, like one group of B cells, become the active one to fight the infection now. Then there is always another group that stays behind. They're called your memory cells. So you have memory B cells and memory T cells. Their job is to just stay put. And they memorize and they remember. So in case you get infected with the same virus, same whatever, you are quick now to respond. That's why if you look at like vaccination, where is that? Right here, look at a primary response to any kind of infection. You see, it takes a while. But look at, if you see, it takes a while. But now if you get an infection again, your white blood cells, they already know what to do. So the response is quicker and more effective. They already know what's coming. They know the type of infection, they can fight it. So you understand that? Both of them get active. It's like one group of soldiers, but half of them will fight the infection now. Half will stay put. They're just there, they remember. That's why they're called memory cells. And that's the principle behind any immunization. Okay, now I want to know, how do they call? How do my second line of defense, how do they call these B cells and T cells? The way they call it is by what you call antigen presentation. Antigen presentation, okay? So, what I want you to know is this term, which I want you to remember all the way to your comprehensive final exam, okay? MHC proteins. Short for major histocompatibility proteins. I'm sure you heard of that many times. Histocompatibility, right? Major histocompatibility proteins, MHC proteins. What you should know is every cell in your body has a name tag. Every cell. Like all my cells they have a name tag, RK. Yours is KB, yours is LG, whatever, right? And the name tag, you remember for your red blood cells, it's only the red blood cells are unique. They have the name tag, the A antigen, the B antigen, you remember that? That's their name tag. For all the other cells of the body, the name tag is called this one. Does it make sense? The B cells and the T cells are also a separate group. We're going to talk about them. But for all the other cells except for this, it's your MHC proteins. So you have two major types of MHC proteins, MHC1 and MHC2. So MHC2 proteins are found in all your phagocytic cells. So all the white blood cells, because they have that property of phagocytosis. So look at this one. And that's how they, I always get reminded of, Lily and I know you know that we love going to the super buffet, right? And I think sometimes it's so funny as we go in, there are some guys who come on, they have a toothpick sticking out, right? That means they enjoyed their meal. So their toothpick is, Lily's trying to copy them with the toothpick out. That's the same thing, you just think that way, okay? So look at, this is a phagocyte, see? It ingested the pathogen, see? And it will try to neutralize it. But it needs to call on the third line of defense. So you see, as it processes the pathogen, look at, it spits out a portion of that antigen, like a toothpick. But it's not in any surface of the cell membrane of the phagocyte, it's sticking it out through it MHC2 protein. Do you understand? That's where it sticks out a part of that pathogen. One more time. This is a phagocyte. Any kind of your phagocytes, right? Maybe your neutrophil, your monocyte, your basophil, dendritic cell, all of those that the eosinophils. So when they ingest the pathogen, this is my second line of defense, right? They have to call the third line of defense. The way they call it is called antigen presentation. And this is it. See? They eat the bacteria, and then they spit it out in like a toothpick. That's the signal. So the B cells and T cells, when they see that, that's how they get activated. That's how they call, see? As they're trying to neutralize it. But where are they spitting it out? It's attached to MHC2 proteins. Okay? All the way to your final exam, I remember asking this. Only because I think it's very important. And I think someone told me that it came out in their board exam. So that's why I want you to remember. Okay, what about the other cells? Right? Let's say my skin cell. It's not a white blood cell. It's infected. So what the skin cells, your infected cells, if it's not a white blood cell, okay? If it's not a white blood cell, your infected cells also, see? Infected body cells, if it's not a white blood cell, they also try to call on your third line of defense. They spit it out through what? Your MHC1. That's their toothpick.
MHC1 is the spitting out?
Yes, MHC1 for the rest of the body cells if it's not a white blood cell. They also have to call. They're helpless. Do you understand? That's what you mean by antigen presentation. You see, they all have to call on your third line of defense. Why? The third line is really the most effective, your best way of trying to control your infection. That's why if your third line of defense is not going to work, then you have to be treated with antibiotics, you see? Or any kind of treatment. If your third line is working, then you're okay. You're going to fight that infection, you're going to win on your own. But once they're down, maybe this is not able to present. If this is not able to present, how can you call? They have to be called. That's the problem. Your B cells and your T cells, they're there, but they have to be called. You see in a minute. You got that? So you have two types of cells, right, that can be infected or that can contain the bacteria or the pathogen. Your own self cells, also the white blood cells that's trying to help you. Both of them are trying to call on your third line of defense. So your own cells, they call it through your MHC1. That's where they spit out signals, which is part of the pathogen. If it's the white blood cell, they spit out signals through MHC2 proteins. Okay? MHC2 and MHC1. You understand? Okay?
So one's the white blood cells and the other one's your own?
Your own cell. If it's not a white blood cell, it's MHC1. If it's a white blood cell, it's MHC2. Okay? How I try to remember 2, so 2, like that. So it's white blood, whereas you can't put a W there. So this must be my own cells, and this is the white blood cells, MHC2. Whatever will help you remember, okay? But don't forget, in your final exam, I remember asking that one, okay?
So the phagocytes call?
That's right.
Okay.
They call on what? Your B and your T cells, which is your third line of defense. That's how we always fight an infection. They're always calling one another.
And then how does that connect
What?
to these two items? I'm confused. You lost me back.
How do they connect to this one? This is how they call. This is called antigen presentation. So again, you have see, there's the pathogen, there's the bacteria. So either a white blood cell, a typical white blood cell, or your own self cell will become infected with the bacteria, right? In the white blood cell, it's trying to ingest it and take care of the problem, but it's going to call on your B cells and your T cells also, bound to MHC2. A typical cell that's infected will just try to spit out. It's not able to process it, but will just try to call on the third line, spitting it out on its MHC1. So whether it's your own cell or your white blood cell, both of them try to call on your B cells and your T cells. And that process, this is what you call antigen presentation. They have to present the antigen, right? If they don't present the antigen, then there's no problem. They have to tell your B cells and T cells there's a problem. How do the B cells and T cells know there's a problem? When they see something abnormal sticking out of you. If there's something abnormal that doesn't belong to your own cell, then that's how they know. Now we call on your B cells and your T cells. Is it clear so far, everybody? Okay, now let's call on our B cells. They got called. Okay? And once they get called, let's look at your B cells first. How do they differentiate? So here, you see, look at the inactive B cell. When they get called, when they get activated, they differentiate into, don't forget this one, okay? They differentiate into plasma cells that make antibodies. Plasma cells are nothing but cells in the plasma. So, the B cells, when they get called, the way they try to fight the infection is by actually making antibodies. What are antibodies? Are they cells? They are not cells. In your comprehensive final exam, you have to be very careful in reading every word. I remember there was a sentence that says antibodies are cells that provide third line of defense. You just automatically write true because you see antibodies and third line. It is not a cell. I just want to know that you know. The antibodies are your globulin proteins, remember?
Oh, yeah. Okay.
Okay?
Mm-hmm.
And that's how they fight the infection. You see? Again, we got to this part, the antigen presentation. You wake up your B cells. When the B cells get activated, you have the active group and the memory group. The active group, how do they fight? They fight by making antibodies. And antibodies are not cells. Where are these antibodies? The antibodies are in your plasma. Because they are not cells, they cannot help you when the bacteria entered your own cell. Don't forget that. Antibodies can only help protect us against pathogens that have not entered your own cells yet. Does it make sense? Because they are not cells. That's why B cells are referred to as providing you with humoral immunity. Humor for fluid. And they protect you against the pathogens that's just outside your own cells. Once those pathogens entered your cell, they're no longer going to be able to help you because they are not cells. Don't forget. So, how do I want you to study these different antibodies? I want you to know the different types, IgG. So there's IgG, IgA, IgM, IgE, IgG. So IgG. Next to IgG, you should write that this is of course the most abundant. This is usually increased if you have an old or chronic infection. Old infection. They're the ones that's given from the mommy to the baby, to the placenta. So while the baby in utero, that's the one that you give to them. That's why the cord blood, they keep that filled with antibodies also. Okay? So that's the one. It protects the newborn. So if your IgG is high, you know that you're fighting an old infection. IgM. If the IgM is high, your infection is a new infection or chronic infection. This is also the culprit why we reject mismatched blood. So ABO reactions. This is the antibody in your blood that will cause that agglutination of mismatched blood.
So that one's chronic too?
No, M is new infection.
Okay, you said and chronic.
Oh, sorry, acute
Okay.
infection. IgG is chronic, sorry. Next, if IgA is the problem, this IgA is present in any kind of secretion. IgA is the antibody that is present in breast milk, in any kind of secretion. This is why breastfeeding is encouraged. Of all the components of breast milk, this is the only one that cannot be added to any formula out there. This is the advantage of breastfeeding. That's why with Lily, I breastfed her until 3 years old. She's never been in the hospital. There's only one time that she had a cough and a cold for 1 day. But other than that, breastfeeding, and this also coats the GI tract of babies. We know that babies, when they're born, they don't have their own immune protection, right? So that's why with babies who are not breastfed, they'll have frequent diarrhea, which is not a common problem. So I remember in the test, I have something about, just always think, if you have frequent respiratory infection or GI tract infection, think IgA. Must have a problem with IgA. Okay? Mucosal protection. IgE. If your IgE is high, in general, you have an allergic reaction or parasite, as usual. How I try to remember, E, like eosinophil. So, same for parasite and allergy. The IgD actually has no actual protection. What they do is, they say it's the one that initiates the immune response, but it's not usually. What people always look at, or healthcare providers, they look at the others, IgG, A, M, or E. They don't look at this one. It has no purpose. Oh, well, sorry, it has a purpose, but it doesn't fight.
So if the IgG and the M and the E's are high, you have some kind of infection, whether it's new or old, but then what if the IgA is high? Is it supposed to be high for the milk, the secretion? Because you said it's present in any type of secretion.
If the IgA is high, then so IgA is supposed to protect you. So if your IgA is high, you must have maybe you have bronchitis, maybe you have diarrhea.
Okay.
Okay, and so this is a summary. So what are they? They are proteins. Can they do anything once that pathogen has entered your cell? They cannot. Don't forget that. All they do is they can prevent the bacteria from entering. How? They do their job this way. You see? Number one, agglutination. You remember agglutination, clumping? So the antibodies, which are not cells, they can clump bacteria together, making them bigger so that your white blood cells, your other cells, can see it maybe, and then they can take care of the problem. Some bacteria are too small, your white blood cells can see it, but once they're big like this, you clump them together, easier to take care of them, right? It's easier to take care of them in groups than individually. So that's one way that your antibodies, your B cells, can help you. Don't forget, these are not cells. So agglutination. Opsonization, you understand opsonization, right? So the antibodies can coat the bacteria, making it more tasty for your phagocytes. They can take care of the pathogen. They can also do neutralization. So antigens have an antigenic surface. So the parts of the cell membrane of your antigens that can harm you will be covered with antibodies, so that even if they're present in your body, they can't harm. You see, they're neutralized by the antibodies that's covering them. You understand this one? You understand this and this, right? They can also activate your complement, the complement proteins. And they can also provide antibody-dependent cell-mediated cytotoxicity, which means, see, they can't do anything. You have your eosinophils, see, this, your white blood cells, these antibodies, again, are not cells, so they can just activate some of your white blood cells. In this case, they're showing your eosinophils. They secrete chemicals that cause lysis of the pathogen. But the antibodies can't do that. They are not cells. I forgot to talk about, I just looked at this one, the natural killer cells. This is another type of second line of defense. Natural killer cells. These are like your phagocytes. But what the natural killer cells do, instead of phagocytosis, they secrete enzymes that can kill. So that's why they're natural killer. It's called lytic enzymes. So they cause lysis of the cell membrane of the pathogen. Okay? You understand this part? What about if the pathogen has entered your cell? Can your antibodies help you? No more. They cannot. So you are SOL, is what they say, right? Here comes the T cells, which gives you cytotoxic immunity. That's why it's called cytotoxic immunity, the T cells. The T cells provide cytotoxic immunity. Whereas the B cells, they provide only humoral immunity. So cytotoxic means they can actually kill the cell. The infected cell even. Your own infected cells. They can kill, take care of the problem. And they can also kill the actual pathogen, which the antibodies cannot do because they're only proteins.
Which is why the HIV virus is
That's
so
very good.
effective.
We're gonna talk about that. Okay, very good. There are two types of cells, okay? Two types of T cells. There's 4 of them, but for our purposes, we're going to concentrate on these 2 types of T cells. Helper T cell and your cytotoxic T cell. Helper T cell. So TH and your cytotoxic T cell. So there are 2 general types of T cells. Sorry, there's more than that. There's 4, but we concentrate on these 2. The the H and the cytotoxic. Okay. When you look at them under the microscope, they all look the same. They're lymphocytes. How do you identify which one is a helper and which one is a cytotoxic T cell? You identify them by their CD markers. CD markers. This is the cluster of differentiation. These are proteins on the cell membrane of my helper or my cytotoxic. Do you understand? Okay? And so this is a typical lymphocyte. If this lymphocyte contains the CD4, it's a helper T cell. See, the number 4 is like a letter H. If this lymphocyte has the CD8 protein, it's a cytotoxic T cell. The number 8 has 2 O's, cytotoxic T cell. That's how I try to remember. Do you understand? Again, there's many other classes of T cells. We concentrate on these 2. I know you've heard of what's the CD4 count? What's the CD8 count? This is what they're trying to count. There's a very important difference between these 2. So, under the microscope, again, they look the same. How do I identify? The CD4, the helper has the CD4 protein on its cell membrane. That's how I identify that it's a helper T cell.
So it just has 4 proteins on the membrane?
No, it's called CD4 protein,
Okay.
not 4 proteins.
Well, how would you know? How do you know it has 4?
What
CD.
for?
How do you
No,
know
it's just called.
That's the name.
It's just called.
How do you know the difference? CD8.
It's by
CD4?
immunofluorescence. So it's a different kind. It's a marker. So they use. I don't know what staining techniques that they use,
Oh.
but they're able to stain the CD4 proteins a separate way, and then the CD8 proteins. When it's the CD8 proteins that stain, then you know that it's a cytotoxic T cell. Okay? Okay, there's a difference between the 2, okay? What is the function? The CD4 or the helper proteins. Just think, helper. They're the ones that shout, help! Okay? You just write, they shout, help. They call on your CD8, your cytotoxic proteins. It is called cytotoxic, if you look at this picture, because it's actually the one that kills, not the helper. So what does the helper do? It calls, but there's something very important about the helper. Okay? This cytotoxic, look at, it's actually the one that will kill, right? Cytotoxic. It will really kill the bacteria or maybe your infected cell. This is the one that does the job. But guess what? It will not do it unless it's told by the helper. And here comes the AIDS. The HIV actually attacks your CD4. You see? So if your CD4 is down, even if this is normal, you're going to die, you're going to suffer. You won't be able to fight an infection. That's why AIDS people or AIDS patients, they die from overwhelming infection, not from the AIDS, right? They can't fight the infection. Why? They may have normal this, but this is the target of AIDS. How can this be called? It's like, they say the husband and the wife. The husband will not do anything unless the wife does.
So the T helper is the person, and the 8 is the gun. Without the person, the gun won't fire.
Yes, very good.
So they can't call the
That is
cytotoxic?
very good, yes. So just think, cytotoxic. This is the one that really kills. But it won't do it. It's like it's God's way of making sure that they get called only if there's an actual you don't want them to keep on doing what if it's not an actual infection. They have to make sure that it's an actual infection. This is the one that calls, you see? That's
so to cure that, we have to figure out how to duplicate a T helper.
how to increase your helper. Yeah.
So is there,
See, helper T cell will activate. Helper T cell will also activate your cytotoxic. Where's that nice
so
slide?
what happens if you, your cytotoxic, proteins or cells don't
What's that?
stop after they've been activated?
What?
What happens if they keep going after they activate the
No,
cytotoxic?
that's very good. They will only be activated if you send out flares or signals. If there's no more signals, then they don't get activated. It's negative feedback. It's amazing, isn't it? It's very interesting. I lifted this from this one. There's another one from Encyclopedia Britannica, I think. But I thought it was nice to help you understand. But you see, all these slides, I just keep on adding. If I see something nice, I add it there. Hopefully, it will help you understand. When you read the book, it's a bit, you think, oh, this doesn't make sense. But if you just picture in your head, see how it's fighting, and you just pick up those terms like helper and cytotoxic, and just know that the B cells, they just make antibodies. So remember, you have, this is your cell. This is your cell. You have the bacteria outside. So there's a strategy. You either want to take care of this before it enters. And the one that will do that can be your antibodies. But once this has entered, you need help. The antibodies can't do that anymore. Your T cells will do that. So the T cells, they're very effective. The cytotoxic is very effective because they can actually kill this. Whereas the B cells will they neutralize it. They can't kill it. They just neutralize it and wait for someone to come, right? The cytotoxic can actually kill this and this. No more. But then this one can only be called by my helper. So now you understand with AIDS patients, don't they always go for regular CD4 count? That's what they always check, the CD4 count of AIDS patients, right? Or HIV positive individuals. They want to make sure that the CD4 count is normal. If the CD4 count is going down, they always advise them, to be healthy and try, I guess, to live in a bubble. And if there's any kind of infection, if that CD4 count is down, they anticipate a severe kind of infection with any kind of HIV or immunocompromised individual. You see why it's very difficult to have a lab exam? Can you imagine if I gave you 3 lab exams? It's too much to study with the lecture part of Bio 242. Okay? So, this is why, now you understand because of the memory, now you understand why we want to do vaccination or immunization. Of course, there's 2 schools of thoughts behind immunization. Some people, what's her name? Jenny McCarthy? She says it leads to autism. So I firmly believe in immunization, but it's all just different schools of thoughts. So the last part of this one, of this chapter, talks about immunity, right? How there's 2 types of immunity, the acquired immunity and your the active immunity and the passive immunity. Just think, when you say active, it means you are actively making antibodies. When you say passive, it's just handed down to you. That's what it means. It's a gift that you receive. So look at the naturally acquired passive and the artificially acquired passive. The naturally acquired passive is the antibodies you get from your mom. It's just a gift. You're given it. You didn't make it. That's what it means by passive. You just receive it. And look at the artificially acquired passive. You're given an injection of antibodies. Okay? Again, passive means you're just receiving it. You didn't have to work. When you say it's active immunity, you yourself have to work and make these antibodies. How do you make the antibodies? When you get sick. How do you get sick? You can either get sick naturally, when you were exposed to, you actually got sick here, naturally acquired active immunity, or you get an intravenous, a vaccination. This is the purpose of the vaccination. So the vaccination is an example of artificially acquired active immunity. Do you understand the difference between active and passive? Active means you have to actually make the antibodies. Passive means you just receive it. You can make the antibodies if you actually get sick. That means you naturally got sick. Or you can make the antibodies if you were given the antigen through vaccination or immunization.
So what's the passive artificially acquired?
Artificially is you just get an injection. See? Injection of antibodies.
Oh. Well, what's the difference between an injection of antibodies for the active immunity? Isn't that the same?
Active immunity?
The artificially
This
active
one?
immunity, I'm confused between the two.
This one?
Yeah.
So active immunity means you make your own. See? You have to make your own antibodies here. You're making it yourself. With this one, you're
it's
just
artificial. How are you making it yourself?
No, what you're given is an artificial sickness, an artificial path. When you get the immunization, aren't they injecting the dead antigen to you?
Okay.
So when you get the dead antigen, you start making your antibodies.
Like the flu shot when you have to.
The flu shot, yes. You're getting as an artificial flu.
Yeah, when you
antigen.
get the vaccine, your body goes, "Ooh, something foreign," and
Yeah.
makes the antibodies. When you
Yes.
get the passive, they're actually giving you the antibodies.
Okay.
They've taken them from somebody else who's already made them, spun them out, and given them to you.
Yes.
Okay. That's
And isn't that done usually when it's too dangerous to give the actual?
Yes. See? Okay? And this is the end of this chapter. So, break. And now I want you to open up our